Hey guys, I hope you all had a great spring break. I wanted to hop on here and go over a little bit of review from last week, just summarizing mitosis, and then we're going to move into our meiosis notes, which is very similar to mitosis, sounds like mitosis, but has a couple of key differences that we're going to have to ensure you know. So, just our main important parts of mitosis that I'm hoping you learned last week. We start off with one parent cell or one somatic body cell that's going to replicate and divide into two identical daughter cells. So there should not be any differences between the parent and the daughter cell. Purpose is here for growth, thinking about going from an infant to adult, repair, if I fall, scrape my knee, and then asexual reproduction, which you could think of as cloning. Usually that's our unicellular organisms making replications of themselves. So the stages here are interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. I remember it as I practiced math at the cafe. Interphase is the longest stage, which has the cell growing. That's where DNA replication takes place. Remembering the enzymes, which I've listed on this slide for you. And then the cell actually physically dividing is going to be that PMAT portion. That's what I say when I talk about mitosis, where the DNA is forming into chromosomes. They line up in the middle, they separate, and then that cell splits. With telophase in a plant cell, you form the cell plate. In an animal cell, you get that cleavage furrow, which will pinch apart. And then the five main forms of asexual reproduction, I want you to know budding, that's usually in a hydra, where a baby hydra will grow off of the side and break off. Binary fission, it would be bacteria splitting into two. Sporulation, if you've ever like kicked a mushroom on the ground and you see the spores fly off, that would be that. Regeneration was the starfish example where its limb would break off and then grow into another starfish, which is not to say go rip off a starfish leg, it doesn't work like that. And then vegetative propagation, if you think of a strawberry plant growing veins and then extending somewhere else to start a new plant. So that's the need to know mitosis. So today we're looking at meiosis, which is basically the cell dividing, but the purpose is sexual reproduction. So, meiosis, Sex Ed 101, we're literally making the process of making sperm cells and making egg cells. So, we're going to look at that today. So, just a couple of things I want to look at. After this, we're going to be getting into our genetics unit. You've probably seen something like this. It's called a Punnett square. And I know you've learned this. You might not have known what these letters on the side mean. And these are actually the father's gametes or the sperm cell. So this could be one sperm cell that has a capital letter. The other sperm cell has the lowercase recessive gene. The mother here has two recessive genes. So this is showing all of the possible offspring from the sperm cells and the egg cells. So we can see different genetic diversities. And then this will come back during our evolution unit to see how organisms have evolved and changed over time. So, just some review of genetic terms. DNA, we know that's our genetic information within a eukaryote, it's inside the nucleus. We know a gene is a small segment of DNA that codes for a trait. If I have my DNA in a loose string like form, kind of like spaghetti, we call it chromatin. So, this would be like chromatin down here. And then once I wrap that chromatin around these proteins called histones, and I form a chromosome, which is very tightly wrapped DNA, which is usually how you'll see it when we're doing the division process. The centromere here holds the two sister chromatids or holds the DNA together. So during metaphase, that centromere gets ripped apart and one of the sister chromatids goes to one side, one sister chromatid goes to the other side. Meiosis is a specialized cell division for sexual reproduction. So the purpose is to make sperm and egg cells so that they're able to unite and form a baby. So we say this produces haploid cells because it's going to have half the genetic information. So thinking about humans, in each of our cells, we have 46 chromosomes. So in order to create that first cell, to create that child, you're going to want the father to have 23 chromosomes and the mother to have 23 chromosomes. So then that sperm and egg meet, that first cell will have 46. That's why it's essential to make that haploid cell or half the genetic information. So you might hear me talk about the haploid cells as gametes. That's the scientific name for sperm and egg cells, basically saying the reproductive cells. So haploid, gametes, those would be key vocabulary terms to know during this unit. So fun fact of the day, during meiosis, males will produce 4 sperm cells within their testes. Females in our ovaries, we actually make 1 egg cell and 3 polar bodies. So those polar bodies will actually be disintegrated back in your body and no longer needed. So one of the main differences we can already see between meiosis and mitosis is mitosis, you're making identical cells with full genetic information dividing once. Meiosis, in order to get half of that information, you can see we're going to go through the cell cycle once dividing, but then we also want to go through it twice in order to get those haploid cells with half the genetic information. So just in case you're on the vocab terms, I put them here for you. You know me with my root words: diploid, 2 chromosomes, the body cells, somatic cells; haploid, half, that's only going to be the sperm or egg cell. So if it's not a sperm or egg cell, you can already say, okay, that's a diploid cell. So we say that the ovary and testes go through reduction division because we're reducing the chromosome number by half from diploid to haploid. So in humans, our diploid cells, 46, 23 of those chromosomes came from mom, 23 from dad, 23 plus 23 is 46. And then our gametes are going to be 23. So this picture down here is the karyotype. We're going to get into this in genetics. So that's just looking at the chromosomes in a human. So just kind of some front loading for you. So what I'd like you to do is pause me quick, and you're going to answer these questions and see if you can identify the correct number of chromosomes. Once you have the answers, hit play and we'll continue. So for number 1, if a horse's retina cell has 36 chromosomes, how many would be found in its sperm? So you should take 36 and we have to divide it in half. So that'd be 18. 2, if N, so N is haploid, 2N would be diploid. So if N equals 4, so I know that's the haploid cell of fruit flies, how many chromosomes does a wing cell have? Was a wing cell used for reproduction? No. So I know I can take that haploid number 4 and multiply it by 2 to get 8. So 8 chromosomes would be in the wing cell. 3, if 2N of a pine tree is 86, how many chromosomes would be found in its pine ovum? So pine ovum is a fancy way of saying the pine egg. So 2N, I'd have to take 86 and divide it in half to get, oh my goodness, can I do math? 43. 4, if N equals 16 for a goldfish, how many chromosomes are in its fin cell? So N, we know that's a haploid cell, so that'd be the goldfish's sperm or egg cell. So I'd have to multiply that to get 32. If you have any questions on that, feel free to email me. Homologous chromosomes, another key term that's just a term for saying, so maybe this is the chromosome for mom, this is the chromosome for dad. You have, as I said, 23 of each. So homologous chromosome is just going to be a chromosome that is the same size and codes for the same trait. So maybe right here on your mom's chromosome, it's the trait for brown eyes. Right here on your dad's chromosome, it's the trait for blue eyes. So although they have different variations, that same gene, that same characteristic is in the exact same location. So here I can just see a homologous chromatid, one from mom, one from dad. Here would be after interphase in the first cycle. So we double that information to prepare for division. So we're just going to quickly go through the cell cycle. I'm not going to do this in depth because you did learn mitosis right before spring break. So just thinking back to mitosis, I practice math at the cafe. So in meiosis, each stage will occur twice. So we have interphase, then we go into meiosis 1, which would be prophase, metaphase, anaphase, telophase, cytokinesis. And then for the second part, notice I skip interphase and I go straight into prophase because I don't want that genetic information to replicate again. So meiosis is going to start with a germ cell. Germ cell is just a fancy way of saying a cell that creates sperm and egg cells. So we'll start off with interphase. We'll go through DNA replication, cell growth, the same thing we saw in interphase. But then at the end of meiosis 1, I should have 2 cells, but I should have genetic information. So let's see how that occurs. So I'm jumping all over the place. So at the start of prophase one, the chromosomes have already duplicated. During prophase one, they become shorter and thicker. And this is going to be the longest stage of meiosis. So I'm going to see my sister chromatids line up. So I call this a tetrad. And you're going to see this thing called crossing over take place, where one chromosome will literally go over the other, and you can see they exchange a little bit of genetic information. So this is how we create genetic diversity. So prophase, one thing of that as the important part of meiosis. If I ask you any phase to know, you should say, oh, prophase one is where crossing over occurs, Mrs. Esther. And I'll say, yes, great. Why do I care about crossing over? And you'll say, oh, well, that creates genetic diversity. And I'll say, oh my goodness, I'm so proud of you. So here's just another image, prophase one crossing over, physically crossing over one another, exchanging genetic information. So then it goes pretty much the same. So metaphase, these guys line up in the middle. Anaphase, they will pull away from one another. Telophase, we'll see that pinch in an animal cell to split into two. Hey, look, that looks kind of familiar. No interphase, we move directly into meiosis 2. So same thing again, that chromatin will wrap tightly into chromosomes. It will line up in the middle. Notice I have less genetic information than the first round, and I have 2 cells dividing now instead of 1. They'll split apart during anaphase. Anaphase, the sister chromatids move away from one another. And then at the end of the meiosis 2, I should end up with 4 haploid cells having half of the genetic information. So as I said before, 4 sperm cells will form for a male. For a female, 1 egg and 3 small polar bodies. So in males, we actually call this process spermatogenesis. And in females, we actually call it oogenesis. So there you can kind of see the process of males versus females, that polar body forming, being dissolved. A couple more just looking at our genetic information. So one way to get genetic information besides crossing over is called the independent assortment, which basically says the traits will be independently assorted from one another. So perhaps, so sperm from the same man and egg from the same woman can have different combinations of genes. So let's say one trait was capital T. So this is just showing all the different combinations. So within the sperm or within the fertilized egg, I could have both dominant traits. I could have a capital T, lowercase s. I could have both recessive traits. I could have lower t, capital S. So it's just showing from these genetics starting off with, once I get that fertilized egg, there's 4 different possibilities. So maybe you have a sibling you look exactly alike. Maybe you're both fertilized the same way, getting the same genes from mom and dad. Maybe you have a sibling where you're like, wow, I don't think they're related to me. Well, perhaps they received more of the recessive traits and you received more of the dominant traits. So that's just showing a little bit of genetic diversity, even in one family. And then the last form of genetic variation that I want you to know is random fertilization. So the random joining of 2 gametes, basically saying that we can't pick what sperm will fertilize which egg. It's very random. So one sperm might have one trait, another sperm might have another. Whoever's the fastest swimmer gets there, and that is who fertilizes the egg. So I believe that's our last slide for today. Let me know if you have any questions, and I hope you all have a great day.